Good afternoon, teacher and classmates. My name is Madeline Mejia, and today I'm going to talk about molecular genetic anatomy and risk profile of Hirschsprung's disease. About the definition, Hirschsprung disease is characterized by a lack of nodes in the mesenteric and submucosal plexuses of the intestine. It is a complex model disorder because it exemplifies multifactorial inheritance and has been molecularly treatable. This disease has a high heritability and the sex difference is marked by the male sex in a 4:2 ratio. About the causes, it has multifactorial causes and various analyses have shown genetic heterogeneity according to the extension of the aganglionosis. The long form is characterized by the autosomal dominant inheritance and the short form by recessive or multifactorial inheritance. The genes involved were the genes encoding the receptor tyrosine kinase RET and the G protein coupled receptor EGNRB.
Today's topic is Hirschsprung's disease, or HSCR. Hirschsprung's disease is a congenital defect that is characterized by the absence of particular nerve cells in a segment of the bowel in a baby. The absence of these cells causes the muscles in the bowel to lose their ability to move stool through the intestine. A newborn who is suffering from this condition usually can't have a bowel movement in the days after birth. The condition is divided into two main parts, short segment and long segment, depending on how much of the bowel is affected. The condition majorly affects the large intestine, or colon, but rarely the small intestine may be affected. Hirschsprung's disease can sometimes lead to a condition called enterocolitis, which is the inflammation of the small intestine and the colon. This is often referred to as Hirschsprung-associated enterocolitis, and is the most frequent complication of HSCR. Enterocolitis can be life-threatening and requires immediate treatment. Severe or untreated enterocolitis can lead to megacolon. Causes. The disorder may occur in isolation or in association with other abnormalities. HSCR that occur in isolation has been associated with mutations in several different genes. Inheritance of these genes can be dominant or recessive, depending on the gene involved. The abnormal genes associated with the disorder can have different effects on members of the same family. A parent who has HSCR has an increased chance of having a child with the disorder. If otherwise normal parents have a child with the condition, the next child has a 4% risk of being affected. The major genes associated with the disorder are the RET proto-oncogene, a gene that encodes for proteins that aid neural crest cells in their movement through the intestines in the developing fetus, and EDNRB gene, that codes for proteins that connect the ganglions to the digestive tract. HSCR that occurs in association with other abnormalities is caused by a chromosome abnormality or genetic syndrome. These may include Bardet-Biedl syndrome, cartilage hair hypoplasia, congenital central hypoventilation syndrome, Down syndrome. People with Down syndrome are at a greater risk. Mowat-Wilson syndrome, Smith-Lemli-Opitz syndrome, Waardenburg syndrome. Symptoms. The most obvious symptom is a newborn's failure to have a bowel movement within 48 hours of delivery. Normally, 90% of babies pass their first stool, or meconium, within 24 hours, and 99% within 48 hours. Other symptoms include green or brown vomit, swollen belly, swelling of the abdomen, excessive gas, bloody diarrhea, explosive stools after a doctor inserts a finger into the rectum. Some cases are diagnosed later into childhood. Symptoms may include fecal retention, chronic constipation, abdominal distension, growth issue, fatigue. Diagnosis and treatment. To make a diagnosis, the doctor may adopt these diagnostic techniques. Anorectal manometry for older children. In this procedure, the doctor inflates a small balloon inside the rectum to see if the anal muscle relaxes. If the muscle does not respond, the child may have HSCR. Barium enema, where the doctor injects a dye via a tube from the anus into the intestine of the child. The dye helps to highlight the inside of the body's organ and see problem areas on X-rays. A biopsy, where the doctor removes a sample tissue from the colon to examine under a microscope. An abdominal X-ray. Treatment. Surgery is thought to be the most effective treatment. This is done in one step or two, depending on how severe the condition is. They include pull-through surgery, where the doctor cuts out the part of the large intestine with the missing nerve cells. Then the rest of the intestine is attached directly to the anus. Ostomy surgery. It is done in two steps. First, the doctor removes the unhealthy section of the large intestine, creates a small opening or stoma in the child's abdomen, and then attaches the top, healthy portion of the colon to the opening. Through this opening, the content of the bowel is eliminated into a special bag. After a time, the second stage, a pull-through surgery, is carried out to close the stoma and connect the healthy portion of the intestine to the anus. Within a year of proper treatment, 95% of people get cured of the disorder. Thank you for watching our video. Please do not forget to like and share the video.
Okay, now we're going to talk about applications in medicine. To assess the role of a gene in Hirschsprung disease, reverse transcriptase polymerase chain reaction, RT-PCR, is used to assess its RNA expression in the human embryonic intestine at carnage stage 22, a time point at which intestinal neurogenesis is completed.
Hirschsprung disease, HSCR, is a complex genetic disorder caused by a mutation in several genes. Despite that in some cases HSCR is inheritable, its exact genetic cause is unknown in about 40 to 50% of the cases. In this study, researchers provided evidence that HSCR inheritance is indeed linked to the mutation of rearranged during transfection, RET, gene and glial cell-derived neurotrophic factor, GDNF, gene. This study was primarily based on a large Dutch family with a history of HSCR. The genetic nature of the disease was identified using human embryonic kidney cell cultures and zebrafish models. In a novel attempt to delete the GDNF gene along with two other inherited variants, IHH and GLI3, the RET activation was reduced, thus proving the major role these variants play in the development of HSCR disease. In the past, the absence of ganglion cells in the biopsy of rectum is used as a method to confirm the diagnosis of HSCR disease. However, the presence of other abnormalities would require patients to undergo genetic counseling to establish the underlying diagnosis further. This study would serve as a reference to greater insights on the genetic mutations involved in HSCR, giving future genetic counseling and genetic tests more accuracy, given the rarity of this disease. Ultimately aligning with the aims of UN SDG 3, good health and well-being, to improve the quality of life by promoting medical researches.
Now about the pros, classical genetic testing for RET, EGNRB, and syndrome-associated genes can be informative, both for clinical research and for risk stratification. Cons, risk prediction for this disease and genetic counseling depend on family history, risk factors such as sex and segment length, and a specific evaluation of syndromic characteristics. In Ecuador, a retrospective case control study was carried out, where 41 patients diagnosed with Hirschsprung disease were analyzed at the Bacartis Pediatric Hospital in Quito between 2002 to 2011. And as results, we have that the pregnancy of those affected was mostly male in a ratio of 28 to 13, with an average age of 2.8 years. And other results about the alleles that we can see in this slide. As conclusion, we have that Hirschsprung disease is usually an isolated condition and not associated with a family history. However, genetic causative factors can be identified in approximately 72% of cases. Therefore, it is important to have a quantification of disease risk according to genotype to address questions of underlying causes and genetic architecture and to provide genetic counseling for patients at higher risk and their relatives. Thank you.